Hello everyone, welcome to Molecura. Today we're going to talk about fructose metabolism. We will talk about the diseases that are associated with fructose metabolism, and we're going to talk about how fructose is metabolized and synthesized in the body when needed. Fructose metabolism is going to be an important topic to discuss. The reason being is that many of the major food sources that we eat is going to have fructose instead of glucose. And this fructose needs to be used by our body to make energy. And how this happens, we're going to be talking about it in detail. The main food source that we eat that it's going to end up being fructose is actually disaccharide called sucrose. So what happens, sucrose that we eat is going to be converted through the action of an enzyme that's called sucrase into two things, and one of them is fructose that comes here, and one of them is glucose. And what we're going to end up with is going to be our fructose that is here. So now our fructose has entered the body and it will go inside the cell. Once that happens inside the cell, this fructose needs to get metabolized to intermediates of glycolysis. And this happens through a couple of reactions. The first reaction that we're going to have is going to be conversion of fructose to fructose 1-phosphate. And we already know this because it's very similar to the first step of glycolysis. And the enzyme that catalyzes this is going to be called fructokinase. And we remember that in glycolysis, this first step was catalyzed by either glucokinase in the liver or hexokinase in other tissues. So it's the same idea here. We are going to be adding a phosphate group here on carbon number 1. And this phosphate group is going to prevent fructose 1-phosphate. Here we have the name, fructose 1-phosphate, from exiting the cell. So once the phosphate group is attached, there is going to be a negative charge. The phosphate group that is attached to our fructose is going to have a negative charge, and this is going to prevent our fructose 1-phosphate to go out of the cell. So it will commit this fructose 1-phosphate to that particular cell. So the next step will be catalyzed by an enzyme that we call Aldolase B. What does aldolase do is that it's going to cut this molecule into half, and it's going to be producing two different molecules for us. It's going to be producing two different molecules. And this enzyme, as we said, is going to be called Aldolase B. Now, the two molecules that are made here, one of them is our famous dihydroacetone phosphate that was there as an intermediate of glycolysis. So the first one is going to be our DHAP, dihydroacetone phosphate. And the second one is going to be our glyceraldehyde. This one is going to be called glyceraldehyde. Now our DHAP can from here go ahead and join the glycolytic pathway. So it will go here to glycolysis. But at the meantime, it can also be used by the cell for gluconeogenesis. So it can go to gluconeogenesis as well. It can go to gluconeogenesis. Now our glyceraldehyde needs to undergo another reaction to be converted into glyceraldehyde 3-phosphate. And glyceraldehyde 3-phosphate is going to be made through the action of an enzyme that is called Triose Kinase. So we have our Triose Kinase here, which is a kinase. And it's going to be converting our glyceraldehyde into glyceraldehyde 3-phosphate. And then our glyceraldehyde 3-phosphate can go, as happened on the other side, can either go to glycolysis or it can go to gluconeogenesis, or it can go to gluconeogenesis. So this is our fructose metabolism, but that's not it. There are other things that we need to talk about here. Before we talk about it more in detail, let's try to also appreciate that in this step we are going to need an ATP molecule. So where this phosphate is coming from is going to be from an ATP, which here is getting hydrolyzed, and hydrolysis of ATP is giving us ADP. So this is an energy requiring step. So the other step that we have here as well, the conversion of glyceraldehyde to glyceraldehyde 3-phosphate, because we are putting a phosphate group here on carbon number 3, we are going to need ATP to get consumed, and there will be production of ADP. So in general, what we are trying to say is that fructose metabolism, until we get the intermediates of glycolysis, it's going to be an energy requiring process. And this is important because we want to shunt the fructose that we have to glycolysis, okay? But as well, during the starvation stage where the body is deprived of glucose, the glyceraldehyde 3-phosphate and our dehydroxyacetone phosphate are going to be important intermediates of gluconeogenesis to be converted into fructose 1,6-bisphosphate, which we're going to be talking about in another video. But before we conclude this, we need to talk about the diseases that are associated with fructose metabolism. So what happens, what diseases that we can have? There are two things that we need to talk about, and the first one is going to be deficiency of fructokinase. So what happens in the deficiency of fructokinase? Here, if fructokinase gets deficient, what happens is that this phosphorylation is not going to happen. We said that this phosphorylation is important to keep the fructose 1-phosphate inside the cell. And without this phosphorylation, the fructose can easily get out of the cell and go to the other parts of the body. So this trapping of fructose inside the cell is going to be defective when this enzyme is not there. Therefore, this condition here is going to be called deficiency of fructokinase, is going to be called as essential fructosuria. Fructosuria. So the increased fructose in the body is going to get excreted through the kidneys and it's going to go in the urine. So this is the condition that is going to be called essential fructosuria, and it's going to be a totally benign condition. However, deficiency of the next enzyme, which is aldolase B, is going to cause many problems. And the reason being is that when this enzyme is deficient, the process goes until here and it gets stopped. So what happens when the process goes until here and it stops is that we are putting a phosphate group on the fructose, and this is going to trap this fructose inside that cell. Now, wherever that fructose is, it's trapped inside the cell. It cannot leave. And that is going to be the main cause of pathology, which is associated with deficiency of aldolase B. This disease is going to be called hereditary fructose intolerance. Hereditary fructose intolerance. And how this disease is going to present, it's going to depend on accumulation of fructose inside various parts of the body. The most important thing is hypoglycemia. After weaning of breastfeeding, this disease is going to become evident because other things like fruits get introduced into the diet, and that of course has a lot of fructose content. So this disease is going to start at that stage. So as we know, a lot of fructose is getting metabolized in the liver. So the first symptom is going to be jaundice, and it's going to be hepatic dysfunction. So the child is going to have jaundice, and which can proceed to become hepatic dysfunction. It can process to fibrosis and all. But these are the initial presentation. So that's the first thing. Second thing is going to be the usual symptoms, like let's say the child will be irritable, there will be nausea, vomiting, and things like that. Along with those things, let's try to write it down. We're going to have nausea and vomiting. Now, apart from that, we are going to have hypoglycemia. And hypoglycemia is going to be an important symptom here, because we need to try to understand why hypoglycemia is happening. Hypoglycemia is happening mainly through three mechanisms. So now we need to talk about why hypoglycemia happens. Hypoglycemia happens because of a couple of reasons, okay? And one of them is going to be impaired glycogenolysis. Impaired glycogenolysis. So we have glycogen in the liver and the skeletal muscle that has been accumulated there by the body. And for this to get broken down into glucose 1-phosphate, what needs to be there is an enzyme that we call phosphorylase A. And phosphorylase A, it's not going to be there because there is depletion of intracellular phosphate. The phosphates are getting put on the fructose, so we have less phosphate here. So what's happening is that our fructokinase is putting the phosphate group on the fructose, and the phosphate is getting depleted, and we are going to have decreased intracellular phosphate. Decreased intracellular phosphate is going to decrease glycogenolysis, because this is going to need an enzyme that is active in its phosphorylated form, that is phosphorylase A. The third point is going to be decreased gluconeogenesis. We are going to have decreased gluconeogenesis. And why is that? Because for gluconeogenesis to happen, we are going to need intermediates of gluconeogenesis, and that is dihydroacetone phosphate, and that is glyceraldehyde 3-phosphate. So we are not having this thing, so gluconeogenesis is also going to get reduced. Furthermore, we are having depletion of intracellular phosphate. So gluconeogenesis and other anabolic pathways are going to need ATP to try to move the reactions forward. We have already talked about how ATP moves reactions that are thermodynamically unfavorable forward. So we are going to have another problem that anabolic pathways are going to be inhibited. And of course, because these are intermediates of glycolysis, needless to say is that we are going to have decreased glycolysis as well. So these are the mechanisms by which our hereditary fructose intolerance is going to cause hypoglycemia. Apart from that, the infant is going to be having nausea and vomiting. They are going to have hepatomegaly. There's going to be jaundice. There's going to be hepatic dysfunction. As the disease progresses, this is going to cause problems in the other organs as well. For example, like the kidney, there is going to be Fanconi-like syndrome happening in the kidney as well. And apart from that, the child will have lethargy, they will have poor feeding, failure to thrive, and this comes with it one by one. So once you see a child with jaundice and there is hypoglycemia, after weaning the child from breast milk and introducing other food types like fruits, that's going to be your clue that this might be hereditary fructose intolerance. And the main clue to the diagnosis is going to be these symptoms, along with hypoglycemia, with taking a careful dietary history. So this is our fructose metabolism. But before we end the fructose metabolism video, we need to talk about what happens during the polyol pathway. So the next pathway that we're going to be talking about is called the polyol pathway. This is called the polyol pathway. And this is going to be something important in the body for various reasons that we're going to be talking about. The polyol pathway is going to start with our glucose. So this is our glucose, and the glucose in the body can get converted into something that we call sorbitol. And sorbitol is going to be an important intermediate, and we're going to be talking about why in a moment. This happens through action of an important enzyme that is called aldose reductase. And we will talk about in a minute why this is important. So aldose reductase is going to use NADPH in this reaction. So there will be NADPH gets used and it gets converted into NADP+. Now, why this is important? Because this is an intermediate in a reaction that's going to end up giving us fructose. So the body is able to take a glucose and synthesize fructose from it. And this is particularly important because there are cells in the body, for example, the sperm, which use fructose as their primary source of energy. So once it's in the male genital tract, it's going to use fructose for energy, but once it enters the female tract, then only it can use glucose as an energy source. So these reactions are going to be important to take the glucose and make fructose out of it. Now, this intermediate sorbitol, it's what can cause pathologies. And we can see here that the name of this molecule as we have is sorbitol. This OL at the end is indicating that this is an alcohol. An alcohol is the molecule that on all of its carbon backbones, it has a hydroxyl group. So these are our hydroxyl groups. On all of the carbon backbones, we have a hydroxyl group, and this is going to make it an alcohol. And it's important because this is going to be an osmotically active substance. An accumulation of sorbitol in any tissue is going to end up giving us pathology. But sorbitol under normal conditions is going to get converted into fructose, as we have talked about. And this is happening through the action of an enzyme that is called sorbitol dehydrogenase. So the enzyme that we have here is called sorbitol dehydrogenase. And sorbitol dehydrogenase again is going to use NAD+, and it will end up giving us NADH. So this is important, as we said, in cells like the sperm to synthesize glucose and use it as energy. So what happens is that some tissues have low amount of sorbitol dehydrogenase. So this reaction is not happening as much as it should be, and sorbitol is getting accumulated. In other conditions, we might have increased glucose, such as diabetes. So this reaction is happening a lot, but this reaction is not happening as much as it should be. Again, because these tissues have lower amount of sorbitol dehydrogenase. This will result in accumulation of sorbitol. And as we said, sorbitol is osmotically active. And this osmotically active substance is going to cause issues. Example of tissues which have low sorbitol dehydrogenase is going to be, let's try to write the examples here. So what tissues are going to have accumulation of sorbitol? It's going to be the lens. In the lens, we end up having cataract. That's why in diabetes, we have a lot of cataract. Other than this, we are going to see this happen in renal tubular cells. In renal tubular cells. And that's why high sugar level is going to give you nephropathy. Another type of cell which has low amount of sorbitol dehydrogenase is going to be the Schwann cells. The Schwann cells of the neurons. And this is going to end up giving you neuropathy. The same thing is going to be true of the retina as well. So these are some examples of tissues which are going to have low sorbitol dehydrogenase. Under normal condition, nothing will happen to them. But under conditions in which glucose is high, such as diabetes, this reaction is going to happen more. You're going to have more sorbitol, but this reaction is not happening as much because sorbitol dehydrogenase is less in those tissues. So the accumulation of sorbitol will cause osmotic damage to the lens, will result in cataract, in renal tubular cells, will result in nephropathy, and in Schwann cells, which will result in neuropathy. By this, we will end our session on fructose metabolism. We will be talking about galactose in the next video. And again, this is going to be an important pathway which we will come back to in galactose metabolism as well, which we are going to see cataract and associated problems.